ALS was the first neurological disease discovered, the first time that physicians could relate the death of a neuron with a specific pathologic process. And what happens in this disease is that people lose the function of their arm or their foot, and it keeps spreading until where they can no longer move a single muscle in their body, and they can only communicate by blinking their eyes. It's generally fatal within 3 to 5 years, though a few patients survive much longer. And we haven't understood the basis for the disease for well over a century. But 20 years ago, the first gene was discovered that causes ALS. And that raised hope that we'd finally begin to understand the disease and start to figure out how to treat it. I was studying how to protect the brain from stroke and oxygen radicals when in 1993 the first gene was discovered to cause ALS. That gene encoded the same protein I was using to protect the brain. I had a theory, though, as to why this protein could go bad and how it would cause neurons to die. We began investigating that process and have been involved in it for the past 20 years. The protein that causes the disease carries a copper atom in it. Our research showed that a lot of the protein was missing copper, so a natural experiment to try was to get copper into the brain. The problem is that copper is quite toxic, and the brain does everything it can to keep copper concentrations very low. So the compound we tested is unique in that it delivers copper very effectively into the brain. One might think you could treat the disease by just taking copper supplements, but that would be a mistake. Copper is very toxic and it does not get into the brain, and it's not going to help. Our own experiments shows that it doesn't work in the mice, and people have taken plenty of copper supplements without any benefit as it is. We're very excited by our success in treating these mice that model ALS. Normally, these mice die in 4 months. We were really excited when the lifespan extended by a few weeks, and then couldn't believe our results when the mice kept living. The mice are now 1.5 years old. Mice normally live to be between 2 to 3 years of age, so these mice are growing old with the disease and still surviving quite well. We know if we take them off the drug that they develop the disease within a few weeks, and we can rescue them by starting the drug again. So, in contrast to taking free copper, the copper-bound ATSM is held very, very tightly and delivered into cells. It's only released in cells that have suffered damage to their mitochondria. We were surprised by how safe the drug is. We were cautious when we initially treated mice with it, but we've not been able to find a toxic level with this particular compound. It's also passed a lot of standard safety tests, the prelude to going to human testing. So it looks very promising for moving to human trials. We're really excited about the prospects of moving this drug into human trials. The response in the animals has a lot of people shocked and in disbelief because no one's been able to develop a drug that's this effective in 20 years. It's by far the strongest response anybody has seen in these mice models ever. We're able to treat the disease after it started and hold the disease off for up to a year. It has great potential for being translated into humans. One never knows if it's really going to work in a human being. There's a lot of safety issues and a lot of other considerations, but we're working through those at this point and working as hard as we can to be able to get this into human patients hopefully within the next few months. So for the 20 years I've been working on this disease, I've watched a lot of patients die, sometimes as rapidly as 6 months. It's really disappointing to see patients die faster than my mice do. I was shocked to find out my daughter was dumping ice over her head, and I learned about the ice bucket challenge through social media. But it was really exciting to see this horrible disease getting the recognition that it deserves. What people need to know is that we've made huge gains in the past 20 years of understanding what causes the disease, and that lets us find new ways to treat it. I'm just enormously excited that we've stumbled upon one approach that worked far better than we dared hope, and I'm praying that it will translate into human studies. We're working with a company called Prosipa to get this drug into the clinical trials through safety, and talking with clinicians about how to properly do these studies. We're also investigating different modifications to the drug that may make it work better, and other ways that might make it even safer or more effective. The other challenge is we do not know if this is going to work in humans. A mouse model is a model. The mouse model we're using is a good model of ALS, and the drug works enormously well, better than any other drug before. But there's no guarantee that it's going to work in humans. So the important thing to understand is it's not just one mouse model we've been successful in treating. We have a mouse model that's the equivalent of being run over by a Mack truck, and it's kept the mice alive for 500 days, and they're still going. We have another mouse model that is much less severe, and those mice are extraordinarily healthy at a year. So this gives us hope that this drug will translate into humans, both for ALS and for other neurological diseases. Investigators in Melbourne, Australia have shown the drug works in Parkinson's models, and there are trials being planned to test this in Parkinson's patients as well as in ALS patients.